Hello, welcome to 13 Universal. This is A-level say biology. This is plant transport P3 series 1, and this is schedule. Let's quickly run through this, okay? Please pick up your pen and your writing materials. Let's solve some questions, okay? So phlegm sap is transported from sources to sinks in phlegm sieve tubes. Each sieve tube is constructed from the phlegm sieve tube element. Okay, you can see that the structure of phlegm sieve tube element is adapted for it to its function. Each of explanations A to F describes how a particular structure features of a phlegm sieve tube element in source is suited to the function of transporting phlegm sap. The matching structural feature for each explanation is listed in table 1.1. The first one, you can see that for entry of sucrose and other organic for rapid of water create hydrostatic pressure. C provides pores to allow the flow of phlegm sap from sieve tube element to the next. D to form very long tubular structure for transport of phlegm sap from source to sink. E decreases resistance to the flow of phlegm sap within each sieve tube element, so the speed of flow is maintained. F provides more space to increase the volume of phlegm sap transported per unit time. So it's complete table 1.1 by writing the correct letter from A to F in the last column of each row, so that each structural feature is matched to the correct explanation. All right, let's see. Okay, yeah, I can see that they use the letter only once. The first row has been completed for you. You can see that. Okay, so they said there is no nucleus. Okay, this has been done for us. Let's go to the end walls are perforated to form sieve plates. So what's the function of this perforation? Let's check. The end walls perforated to form sieve plates. Okay, which one? For entry of sucrose or organic. Okay, for rapid. Now the end walls is between two. Pause the video and try to answer it. Okay, all right. So you see that end wall allows the pores allow the flow of phlegm sap from one sieve tube element to the next. Do you get? Yeah, from one sieve tube element to the next. If you remember how we do this, Yeah, see. Okay, these are phlegm sieve tubes. You can see that. See, these are end walls, perforated end walls. Okay, that allows the movement of phlegm sieve from one to the other end, one sieve tube element to the other sieve tube element. You can see that. All right. Yes. So it moves from here to here to here. Okay. So that's why C, all right, goes with that first one. See this one. So this should be C. The end walls are proposed to form sieve tubes. That's C. Okay. All right. Let's quickly go back. Okay, we didn't check it. Let's check the next one. The next one says there is only a thin layer of cytoplasm around the edge of the cell. So why do we have thin layer of cytoplasm? Why do we have a thin layer? All right, we have a thin layer for each of these thin layer. Okay. Why? Okay. I think that's for to decreases resistance to flow of phlegm sap within each sieve tube element, so the speed of flow is maintained. So that creates more room, reduces resistance to the flow. That's why we have thin cytoplasm there. Is that okay? So that's E. So those should be E. The next one says the cell is elongated and arranged end to end with other cells, end to end with other cells, end to end. Like I drew it here, end to end with other. Why is it elongated? So they join end to end with other cells. Okay. So this says provide more space to increase the volume of phlegm sound transmitted per unit time. Yeah, this makes sense. Is there another one? I suppose the video and try. Provide pause for rapid. No, I think it should be. No, this has been taken already. All right. Decrease resistance. Okay, yeah, that's the to form very tubular. Yeah, this is going to form long tubular structures. Okay, for transport of phlegm sound from source to sink. So from source to sink, long tubular structure. Okay, that is why they are joined end to end to form long tubular structure. So that's D. D is this one. Okay, this is D. The cell has plasmodestic matter connecting to a companion cell. So what is the role of plasmodestic matter? Okay, remember that, guys. Remember, this is very critical. Okay, remember that companion cell is usually associated with sieve tube element, and then we have the sink cell, sorry, source cell. Okay, so if you check it out, you see that basically there is plasmodestic matter between, this is companion cell, this is source cell. So sucrose is loaded from here to here by companion cell. So but when it gets into companion cell, it moves through plasmodestic matter. Okay, so the function of that plasmodestic matter is that we have chosen this. Okay, A says, okay, for entry of sucrose and organic compound. Yeah, that's correct. Okay, yeah, so that they can move from companion cell into the sieve tube element. And of course there is thin cellulose cell wall. Okay, so you see this is loaded from here to here. Why do we have thin cellulose cell? Of course, for rapid entry of water to create high hydrostatic pressure. So once sucrose is loaded from companion cell here into the sieve tube element here, it lowers water potential and then water moves in, okay, for neighboring cells to increase hydrostatic pressure. Okay, so guys, let's see what the mass scheme says. Okay, see that? So it should be C E D A B. Okay, so check it out and ask questions. So at the source, at the sink, sucrose and other organic compounds are unloaded from phlegm sieve tube element. Explain why the process of unloading helps the mass flow, okay, of phlegm sound from source to sink. Why is the process of mass flow helps in the process, guys? Remember, if this is just to mention, okay, if this is a phlegm tube, right? But the phlegm tube consists of the phlegm sieve tube elements join end to end, right? Join end to end. They are joined end to end. The sieve tube elements are joined end to end. So you can see now, but you see, this is the companion cell at the source, and this is of course the source cell where sucrose is made. This is sink cell, right? So this is that's companion cell, and then the sink cell. You can see, guys, pardon me, right? Apologies. Now look at this. So you see that sucrose is loaded into the sieve tube element at the source, and once it's loaded, water will move it because it will lower water potential. So water moves from neighboring cells into here. So once water moves, the water potential increases, and then of course once it increases here, hydrostatic pressure will increase. So that's how hydrostatic pressure is set up. The question is asking us to explain why the process of unloading. So unloading occurs at the sink. So at the sink, sucrose is moved out. So sucrose is moved out of sieve tube element here into the sink through the companion cell. So when sucrose is moved out, what happens is that it increases water potential, right? So water therefore moves out by osmosis. So when water moves out by osmosis, the volume of assimilates decreases. Therefore, the hydrostatic pressure here, decreases. So that sets up hydrostatic pressure gradient between the sink here and the source. Remember, the source already has high hydrostatic pressure. This has low hydrostatic pressure. That sets up. So the movement of sucrose out of the unloading, right, allows mass flow because it creates low hydrostatic pressure. I've said it. It lowers water, increases water. Once it moves out, water potential increases. Water moves out, and then that lowers water potential, creating low volume, all right, and low hydrostatic pressure, which sets up hydrostatic pressure gradient. So you get, look at it. Water leaves sieve tube element, follows sucrose. So sucrose moves out, water moves also with it. All right. So down water potential gradient from high to low. That's what happens, of course. So what happens? That decreases the volume in sieve tube, and once volume decreases, then hydrostatic pressure also will decrease, and then pressure in sieve tube at source, all right, higher at source than sieve tube, and that maintains pressure gradient for the source to sink. So sap moves down pressure gradient. So the movement of sucrose creates low hydrostatic pressure, while the movement of sucrose in here, loading increases high hydrostatic pressure, unloading creates low hydrostatic pressure, and we have to explain that. Okay? Once you unload, water potential increases, water leaves also, right? Volume decreases and hydrostatic pressure decreases. Once you load in here, water potential decreases, water moves in, all right, hydrostatic pressure, volume increases, and the hydrostatic pressure increases. And due to the hydrostatic pressure gradient, high here, low at the sink, high at the source, so Smith moved by mass flow from source to sink. Okay? Alright. So let's quickly. The veins of a leaf contains transport tissues. Okay? Fig 2.1 is a drawing made from an electron micrograph photograph showing a cross section of the transport tissue in a leaf vein. The cells labeled A are modified companion cells known as phloem transfer cells. Transfer cells move sucrose and other assimilates from mesophyll cells B into the phloem sieve tube element C. So let's look at. So that's it, guys. So this is A. A is called companion cell. Can you see? It's actually companion. It has a lot of mitochondria to provide ATP for pumping of protein into cell, right? Okay, as nucleus. And B is actually mesophyll cell. Okay, where's that B? Can you see? You see what organelles are these? Okay, chloroplast. Yeah. Okay, so you can see that. Let's go. It says state the functions of the mesophyll cells B and sieve tube element C and explain how their structures adapted to their function. I want you to pause the video, right? So cell B, okay, function. So you go to cell B. What is the function of cell B and what's the adaptation? All right. So look at cell B. This is B. That's B. That's B. That's B. Okay. So pause the video and try it. The function of B, mesophyll cell, is photosynthesis. Okay. So it actually makes sugar by combining water and carbon dioxide in the presence of light and chlorophyll. Okay, so what adaptation does it have? So write down what adaptation. Write it, pause the video and try it. Can you see it has chloroplast? Chloroplast contains chlorophyll, which traps sunlight. Okay. All right. So you see that pushes chloroplast, also large vacuum pushes chloroplast to the edge to absorb more light. Is that okay? Yes. So, all right. So that is it. Let's check it out. The next one, cell C function adaptation. So, what is the function of cell C? Can you cell C? What is the function of cell C? What is the function of cell C? Remember that cell C, all right, is already called phloem sieve tube element. So it transports, assimilates from source to sink, or it's responsible for translocation. That's what phloem sieve tube element, they're involved in translocation, right? So, but what adaptation does it have? Okay, You can see that it has, this is plasmodious matter, that's plasmodious matter. Okay, why does it have plasmodious matter? Okay, plasmodious matter allows the loading of sucrose from companion cell. This is companion cell, that's companion cell, right, into the sieve tube. Okay, if you check, it has fewer organelles. That reduces what we call resistance to the transport, all right, of assimilates. Okay, there are a lot of features. It has sieve plates which provides support, and then the sieve plate has pores which allows movement of sucrose from one sieve tube to another. Okay, all right, let's see what the mass can say. Okay, photosynthesis for cell B, conversion of carbon dioxide to sugars using light energy, synthesis of carbohydrates. You see, a complex organic compounds from inorganic, right, using light. So what adaptations? You can see adaptation. Large vacuole. First of all, chloroplast absorb light. Large vacuole keeps chloroplast at the periphery to absorb more light. Yeah, adaptation, it has starch grains which stores product of photosynthesis. Large moist surface, alright, cell wall evaporation for water transpiration. Provides water for photosynthesis. See, thin cell wall gas exchange. Alright, see the shape. Okay. Now cell C, sieve tube, cell sieve, you can see that transport or movement of named sugar assimilates, okay, involved in translocation, accept translocation. All right, you can see that. So at the edge, thin layer, so it has thin layer of that's peripheral cytoplasm at the edge that allows maximum volume space of transport. Okay, so the feature here is that it has peripheral or thin cytoplasm at the edge, and that allows maximum volume transport. Okay, less or little resistance. So no nucleus of few organelles allows maximum volume. The same thing, less resistance. Elongated to make sieve tube element. Okay, yeah, so it's elongated. Plasmodious matter helps in unloading of components from components, right. Sieve pores, you can see that flows, right. So these are the features of sieve tube elements, right. These are the role of those features, okay, that allows it to carry out translocation. The cell wall of transfer cells A, shown in Fig 2.1, have infoldings. Explain the advantages of these wall infoldings for the movement of sucrose from mesophase cells to phloem sieve tubes. So what are the advantages? Look at the infolding there. Look at it. We're going back now. Okay? Yeah. This is infolding. Can you see this infolding? So pause the video. What is the infolding? The advantage of infolding. So you see the advantage of this infolding provides large surface area. Is that okay? Anywhere there's infolding here in biological structure, it provides large surface area. That means that it therefore increases the number, okay, of transport proteins. The transport proteins here, we have proton pump, which helps to pump proton. Okay. So guys, so you know that hydrogen ions are pumped from here, cytoplasm of companion cell, to the cell wall to set up proton gradient, and then protons then move back with sucrose through co-transporter. So this infolding increases the number of hydrogen ion pump. That helps to set up more proton gradient, okay, and then it contains more co-transporter for sucrose and hydrogen ion. That means it allows more sucrose to, all right, be loaded. So more sucrose moves into the companion cell, all right, and then moves to sieve tube elements, all right. So those two transport proteins are very important. Okay, so let's look at what the oracle says for that question. Okay, you can see. So increase surface area, volume of cells increases surface area, cell membrane. So the idea of more space, okay, for proton pumps, so carrier proteins, and that, so pumping protons from cytoplasm into cell wall, okay. Yeah, so creates proton electrochemical gradient. That's why we have more proton pumps. So we're going to have, you know, electrochemical gradient or concentration gradient setup. So idea of more space increase for more co-transporter carrier protein. So this co-transporter carries both hydrogen ion and sucrose. Okay, so co-transporter can see that. Yeah, see the function. So more sucrose transporter, right? Okay, just 3 marks, guys. Can you imagine? Fig 3.1 is a diagram of an area of phlegm tissue from a transverse section through the stem of a squash, Cucurbita pepo, cubita. You can see that. All right. Says cell X and cell Y in Fig 3.1 are sieve tube elements. These guys are sieve tube elements. Explain why cell X and cell Y have a very different appearance and structure. So you can see that this is sieve plate, right? This is just the lumen. So that means that cell Y is higher. The height of is at a higher height than cell X. Okay, because sieve plate is the end of a sieve tube element, right? So if we are cutting it here, that means that it's somewhere below, all right, than Y. So Y is at a higher position. Is that okay? Don't forget that if this is it, right? Watch it, my guys. Right? Sieve plate. Watch if this is sieve plate here. Okay, this is sieve plate. Right? So possibly. Sorry. Let me clean it up. Let me clean this. All right. Yeah. So look at this, guys. So we have so this is where cell Y is here. That's sieve plate. Okay. Cell X is somewhere here. That is where the lumen is. So cell Y is higher. All right. Is at a higher position than cell X in the stem. Okay, so you can see that cell Y shows a sieve plate. I did a sieve plate at different height. This is higher, this is higher, this is lower. Okay, and the stem section, right? So stem section misses plates in cell X. So that means we miss the plate. That's another name. Alright, sucrose is formed from the glucose synthesized by mesophase cells in leaves of C. pepo. Right? Explain how companion cells are involved in the transport of sucrose into the phlegm sieve tube. So I want to pause the video and try it. I think we've explained it. That companion cell, pause it. Companion cells contain, they help in loading of sucrose. That's what they do. Right? They load sucrose. They load sucrose. How? Okay, they say, why do they load sucrose? Of course, they have hydrogen ion pump, which pumps hydrogen ion from its cytoplasm to its cell wall, and then set up hydrogen ion gradient. So hydrogen ion moves in, okay, through a co-transporter. Hydrogen ion is moving in by facilitated diffusion. Why? Okay. Why sucrose moves in by secondary active transport? Okay, they pass through the co-transporter. So both of them pass through the co-transporter, and the one they enter, all right, from the sieve tube element, sucrose moves in, all right, through the plasmodic matter. From the companion cell, it moves into. Now I'm going to show you that later, but look at it. I'll show you the illustration. Okay. So you can see that companion cell surface membrane pumps proton into cell wall and then uses ATP, that's active transport. Why does it do that? To establish a proton gradient. So this is proton. Proton moves into companion cell by facilitated diffusion, but that is moving down with the electrochemical. Okay, so proton is co-transported, right, together with sucrose. Sucrose moves against concentration gradient, that's secondary active transport. Okay, All right, so that is the role. Just 4 marks, guys. Hydrogen bonding is important in movement of water in xylem. Explain how hydrogen bonding occurs between two water molecules. Okay, how does it occur, guys? All right, hydrogen bonding, how does it occur between two water molecules? Remember that oxygen, look at this is a water molecule. All right, water molecule has hydrogen. Yeah, this is hydrogen, oxygen of the water molecule. They're all joined by covalent bonds. Okay. All right. This is another water molecule. Right. This is oxygen of that water molecule. All right. I know you know this, but you see, because of dipole, what happens is that hydrogen has small positive charge because oxygen draws more of the electron to itself, so it has small negative charge or partial negative. So you can see this is partially the same thing, right? Partial negative, right? So there's an attraction between hydrogen bond is formed between the hydrogen of one water molecule and oxygen of another water molecule. You can see that, okay? You can see that here. We have this. You see that hydrogen. Can see there's an attraction between hydrogen of a water molecule and oxygen of another water molecule. That's how it is formed. Okay, because it has small negative charge, it has small positive charge. No, so this is small negative and small positive charge. Okay. All right. This could be answered that you can show the diagram. See the diagram there. You can see. So this is a water molecule. This is another water molecule. The hydrogen of this has small negative charge. So that's oxygen of this water molecule. Why the hydrogen of this water molecule has small positive charge, and that's the hydrogen bond between them. So idea that oxygen has a small negative charge, hydrogen has small negative. The hydrogen bond forms between the oxygen of one water molecule. This is oxygen of one water molecule and hydrogen of a neighboring water molecule. That's how it's formed. Okay, oxygen is more electronegative than hydrogen. That's why it's more negatively charged. Is that okay? All right. Outline how hydrogen bond is involved in water transport in xylem of a plant cell. So you see, hydrogen bonds form between two water molecules. Sets up what we call water column. So if this is xylem, right? Don't forget that. The first thing is that hydrogen bond, sorry for the background noise. I'm in a school environment. Students are doing extracurricular, right? So let's assume that these are water molecules, okay? So you can see water molecules. So water molecules stick to each other. So hydrogen bond is formed between water. This is hydrogen bond. So hydrogen bond, that's due to cohesion. Hydrogen bond is formed between water molecules. And that sets up what we call water column. So water column is created by hydrogen bond between water molecules. The hydrogen bond also form, water molecules also form hydrogen bond with the wall of xylem. Okay? Now the wall of xylem has cellulose, alright, forms hydrogen bond, is polar, forms hydrogen bond with water. Okay? So look at that. So this is another hydrogen bond forms wall of xylem. So that's adhesion. So adhesion between water molecules and wall of xylem, hydrophilic part of lignin, okay, helps to maintain the water column. All right, but you see the transpiration pool helps to pull the water column up. Okay, so now guys, watch. I want to show you something here. Watch this. Let me show you this. Assume that this is the leaf, this is the root. So I see that the water column is set up. How is it set up? Hydrogen bond between water molecules due to cohesion set up this water column. Hydrogen bond between water molecules and the wall of xylem helps to maintain the water column. Now, as water molecules at the top of xylem leaves the xylem, all right, to replace water lost by transpiration, it pulls other water molecule beneath it. That's cohesion tension. That's transpiration pools. Transpiration pool helps to move water from water column from the root to the leaf. Okay, so look at it, guys. So cohesion between water molecules, right? Addition hydrogen bond between water molecules, cellulose, hydrophilic bond. Okay, formation of continuous column. So it is that forms continuous column. Hydrogen bond between water molecules maintained by adhesion, and then column pulled by transpiration. So transpiration pull is what pulls it up, guys. All right, let's move to the next one. Hydrogen bonding between water molecules gives water a relatively high latent heat of vaporization. So just why it is important to plant that water has a high latent heat of vaporization. Why does water have, okay, what is the role? Pause this video and try it. Okay, remember that latent heat of vaporization, that is the heat, right, that is needed to convert water from liquid to vapor. Okay, so that means a lot of heat is needed to convert water from liquid to vapor. That is, when evaporation occurs, it leads to cooling effect. Evaporation removes excess heat from plants, okay, thereby preventing enzyme denaturation. Guys, look at it. All right, so plants heated up, okay, you see, plant leaves heat. The light absorbed by photosynthesis heats each plant. So latent heat variation leads to a large cooling effect, so or removal of large quantities of heat, energy by evaporation, and reduces protein or enzyme denaturation, reduces rate of water loss by transpiration. Okay. All right. Okay. Xylem and phloem are the transport tissues of plant. Both tissues have more than one cell type. Okay. The conducting cells of xylem contain xylem sap, and those of phloem contains phloem sap. Okay, composition of xylem sap differs from composition of phloem sap. Yeah, let's look at it. Explain why water is the main component of phloem sap and xylem sap and phloem sap. Okay, so water is the main component of phloem sap. Why? Because water helps due to adhesion. Anyway, why am I solving this? Trying to solve it, okay? I want to pause the video and solve it. So due to adhesion, all right, or due to cohesion between water molecules, like I've explained in the previous question, okay, water helps to form continuous column, right? Okay, which also helps to transport, which helps in water transport, okay? Water column is formed due to adhesion, cohesion between water molecules, and due to cohesion is maintained, and that helps to move water up xylem from the root to the leaf. So water has a behavior, cohesion and adhesion. All right, that's why it helps in water transport. Okay, that property. Now phloem sap, water also helps. All right, remember that in phloem, okay, due to increase in water potential at the source, so we have high hydrostatic pressure. So water, increase in water, all right, sets up hydrostatic pressure gradient in phloem. So we need water is needed to set up hydrostatic pressure gradient, so that, you know, of course, in the source, at the source, we have more water potential because water moves in due to loading of sucrose. So we have high hydrostatic pressure at the sink. Because of unloading of sucrose, we have less water. Okay, so we have low water potential and then low hydrostatic pressure. So look at it here. So can you see? Water is a solvent for assimilates. So it's a solvent, that's number one. Polar molecules. Now, substance in xylem and phloem are transported in dissolved water. So they transport minerals, whether it is in xylem or whether it is in phloem sap. Okay, but look at, for features of xylem, water can move up as part of transpiration. Okay, cohesion between water molecules can move up, adhesion between water molecules, hydrogen bonds, right? Features of phloem, water used to build up hydrostatic pressure gradient. Okay, this is what water does. All right, so you can see. So xylem sap, water moves down. So that is it, guys. Look at it. 3 types of cells associated with translocation sucrose. of sucrose are companion cells, mesophyll cells, phloem sieve tubes. A, B, and C are three events that occur at the source. Each event refers to the cell, but does not name the type of the cell concerned. A, sucrose moves through plasmodious matter into the cell. Hydrogen ions are transported out of the cell. Sucrose moves into the cell through co-transporter protein. Look at it. So this is A. So let's look at the question. Complete table 6.1 by matching the event with a correct cell type. Each cell type may be identified once or not at all. So A, which cell is A? This is event. Okay, sucrose moves through plasmodious matter into cell A. Okay, this cell A is what? I want you to attempt the answer. Cell A is what? Cell A is sieve tube element, because sucrose moves from companion cell through plasmodious matter into the sieve tube element, right? Now B says hydrogen ions are transported out of the cell. So what is cell B? Okay, what is right? The answer. Cell B is companion cell, because hydrogen ions are moved out of companion cells into the cell wall, cytoplasm components into cell components. Okay. Now cell C says sucrose moves into, statement C says sucrose moves into the cell through co-transporter. Okay, it is the same word. Write it down. That's of course companion cell. So sucrose moves back through companion cell, right? Yeah. Look at 3 marks. Okay, phloem sap is transported within phloem sieve tubes. Monosaccharides and disaccharides are transported as part of phloem saps, but polysaccharides are not components of phloem sap because they are heavy, polymers. Define the term disaccharide and polysaccharide. I want to write it down. Okay, try this. So disaccharide consists of 2 monosaccharides joined by glycoside bond. Polysaccharide consists of many monosaccharides. Look at it, yeah. That's like a 2 monosaccharide units. Polysaccharide more than 2. Okay? Yeah. So that is it, guys. Let's look at this. Sieve tubes are composed of phloem sieve tube elements. Fig 4.1 is a diagram of one sieve tube element and its companion cell. Okay? And the structural of the structural features of the sieve tube element have not been included. Okay, there's some of the structural features are not included. Right? See it. So the question says, complete Fig 4.1 by drawing and labeling the structural features of the sieve tube element that have not been included in the diagram. So complete it, guys. I want you to pause this video and complete it. Right? So remember, we can complete this sieve tube. All right, this is sieve plate. Sorry, see, I'm completing it now. Sieve plate, and with pause. Okay, you do the same thing at the other end of the cell. Okay, so let's complete it, guys. Sieve. Okay, so you can see the pause there. So this is sieve plate, but it has pores. Sieve plate is perforated. All right, end walls. Okay, I can just add. Now this is thin cytoplasm. Thin cytoplasm. Okay, you can see thin cytoplasm. Show it. Thin cytoplasm. I'm showing thin cytoplasm. Sorry, let me wipe it off. Okay. Yeah, the mistake somewhere. Thin cytoplasm, you can see that. Okay. Right. Then I can also add mitochondrion. Can you see? Okay. Or any other organelle like smooth endoplasmic reticulum. But I shouldn't add nucleus because it doesn't have nucleus. Okay. All right, let's look at what my scheme says quickly. Yeah, my scheme says if large vacuole or nucleus drawn, okay, maximum. Okay, says sieve plate drawn and labeled. This is sieve plate drawn, you have to label it. Okay, cytoplasm labeled and drawn as thin layers. This is cytoplasm thin layer. Okay, thin layer, you have to label it. Mitochondrion drawn and labeled. This is mitochondrion. Okay, this is SER. Okay, cell surface membrane drawn, you just draw it, and all of these other, all right, proteins. It says a pressure gradient in a sieve tube causes the mass flow of phlegm sap from the source to the sink. At the source, a decrease in water potential in the phlegm soup and an increase in the hydrostatic pressure of the phlegm sieve can be measured. Describe the event that causes each of these changes in the phlegm sap. I want you to describe what causes decrease in water potential, right? Yes, water, when sucrose is loaded into the sieve tube, right? Sucrose is a solute, so that decreases water. So movement, loading of sucrose from the companion cell, all right, into the sieve tube element decreases water potential. Now, increase in hydrostatic pressure is as a result of water moving from neighboring cells by osmosis. Water moves into sieve tube, all right, by osmosis, that increases it. I can see that. Active loading entry, sucrose enters, increases this, right? Water moves in by osmosis. Down water potential. Okay, so guys, thank you for hanging on. Please follow our videos, share, like. So see you next in the next video, which will include transporting animals and the rest. Okay, bye. This is Shedro. Bye bye.